Morning, good afternoon, good evening, everyone. You are welcome to today's lecture. In today's lecture video, we are going to be discussing about enzymology 3, which under this we will be discussing about enzyme inhibition, the effect of enzyme inhibitors on Langmuir-Burke plots, regulation of enzyme activity, and clinical applications of enzymes. So by way of introduction, we can say that for several compounds that we can see as pharmaceutical products or drugs that are being produced by pharmaceutical industries, they act as enzyme inhibitors by inhibiting the activity of the enzyme. And the study of enzyme inhibitors has provided insight into the mechanism and pathway of catalysis. And for example, the types of functional groups that are found in the active site and the role they play by these functional groups, the role they play in the enzymes, in the proteins. So these enzymes, they are most of the majority of these enzymes they are protein nature, and they have what we call active site. And active site is where catalysis occurs. In the active site, there may be a group of amino acids that comes together to form the active site, and that is actually where the catalysis, the process of converting the substrate to product, actually takes place. Because those active site residues, they have functional groups that help in the process of enzyme catalysis. And so we see that most pharmaceutical industries, they design compounds as enzyme inhibitors. So we have two different types of enzyme inhibition. We can say we have reversible inhibition or reversible enzyme inhibitors. If we say we can divide the enzyme inhibitors into two, or the inhibition into two. So we have reversible inhibition, and for the inhibitors, we can say we have reversible inhibitors. And the second one, we have irreversible inhibition, and we have those compounds that act as that serve as irreversible inhibitors. We call them irreversible inhibitors. So this actually can be distinguished by checking the effect on the Langmuir-Burke plots. You remember in the previous class we discussed about Langmuir-Burke plots, right? We talk about Michaelis-Menten equation, and that's where we talk about the plots, the various plots, Langmuir-Burke plots, Eadie-Hofstee, and so on. So some of the enzymes, they play regulatory roles in metabolism. That is to say, they act by controlling the activities of other enzymes. So as some enzymes, they have what we call allosteric site, and those enzymes, we call them allosteric enzymes. They have, in addition to the active site, they have other binding sites that is not the active site, and that is mainly for regulation, for the controlling of the activities of the enzyme. So we call them allosteric enzymes. We have covalently modulated enzymes, and which we say, and they are coenzymes, and we can say that enzymes are very useful tools in clinical diagnosis. So let's look at the inhibition. We said we know the basic knowledge of metabolic pathways is determined by using activities of enzyme, and we can use specific enzyme inhibitors to carry out those processes. So the study of enzyme inhibition, it gives us a valuable information, and that has been obtained in on the mechanism and pathway of enzyme catalysis and substrate specificity of enzymes, the nature of functional group at the active site, and the participation of functional groups in maintaining the active conformation of the enzyme molecule. So pharmaceutical compounds, usually drugs, are mostly designed as inhibitors of specific enzymes. Okay, so let's say, for instance, for a particular disease, there may be maybe a pathogenic parasite or a pathogen that is causing the disease. Let's say, for instance, let's say malaria. So if we the pharmaceutical industries may try to target a particular enzyme in the metabolism of the parasite, so that if that enzyme is very essential or critical to the metabolism of the parasite, it can be targeted. So a drug can be designed so that when that enzyme is inhibited in the parasite, then the parasite will not survive and the parasite can be killed. For example, let's say penicillin. It works by preventing completion of cross-linking between two adjacent peptidoglycan chains in the cell wall of the bacteria. So when this bacteria is carrying out its normal metabolic activities, they produce what we call peptidoglycan layer in their cell wall, which is very important in maintaining the integrity of the cell wall of the parasite. So penicillin, what it does, it goes and inhibits the activity of glycopeptide transpeptidase, which is the enzyme that carry out the process of cross-linking the peptidoglycan chains in the cell wall of the bacteria. So penicillin now go and arrest the activity of that enzyme. Okay, so now if that enzyme did not carry out the activity, it means the parasite will not be able to synthesize the cell wall, and so the cell wall integrity or the bacterial integrity will be compromised, and by so doing, it means the bacteria will not survive. So that we have another example, disulfiram, which is a drug that is used to treat alcoholism. So how does this work? So, in alcohol metabolism, it is carried out in two steps to form acetic acid. So the first enzyme is called alcohol dehydrogenase, which catalyzes the alcohol to bring acetaldehyde, which is then converted to acetic acid by aldehyde dehydrogenase. So the later enzyme, which is aldehyde dehydrogenase, it has an active site cysteine residue that can be irreversibly modified by disulfiram, so resulting in the accumulation of alcohol and acetaldehyde in the blood. So people who take uh, their sulfuram, they become sick because of the accumulation of acetaldehyde in the blood and tissue, which leads to alcohol avoidance. So we have what we call also NSAIDs, uh, NSAIDs that stand for non-steroidal anti-inflammatory drugs. These are drugs that serve as anti-inflammatory activity, like aspirin. Aspirin, it works by inhibiting cyclooxygenase. Okay, it works by inhibiting cyclooxygenase activity, and it's a kind of reversibly block the channel for arachidonate in the enzyme. So uh, we say compounds that inhibit cyclooxygenase, they have anti-inflammatory activities. So drugs are designed to inhibit enzymes that are unique to a particular microorganisms, which will produce few side effects in patients. For example, we have sulfur drugs that are used because they inhibit folic acid synthesis in bacteria. So let's say sulfur drugs can be used as antibiotics. They are used as antibiotics because they target a particular folic acid biosynthesis in the bacteria, which in humans we don't have that biosynthetic capacity to produce folic acid, so it will have just few or no side effects on us. So that is actually one of the importance of checking for the activity of a particular enzyme in both the parasite and also humans. So if you are targeting a particular enzyme as a pharmacist, or you are trying to design a drug, you will design a drug to kill the pathogen, but it should not have effect on the patient. Okay, so you have to check whether to see whether that drug can also have effect on the patient. Maybe if humans have the same enzyme with that parasite, so you need to be very careful in designing inhibitors. So we have three major types of reversible enzyme inhibition, which are competitive, uncompetitive, and non-competitive inhibition, and they can be distinguished by checking the effect of the inhibitor on the reaction kinetics of the enzyme, which may be analyzed in terms of basic Michaelis-Menten equation. So we have competitive inhibition. In competitive inhibition, simply what it means the inhibitor is competing for the active site of the enzyme. You know, the enzyme has a specific substrate that it catalyze, right? So the competitive inhibitor will also come and compete for that binding site that the substrate is supposed to bind. Okay, so we have a competitive inhibition. Instead, it competes with the normal substrate for binding at the active site. So it reacts reversibly with the enzyme to form what we call enzyme inhibitor complex, EI, which is similar to enzyme substrate complex if the substrate is able to bind to the enzyme. You remember? So we have enzyme plus the inhibitor, we have enzyme inhibitor complex. So a competitive inhibitor, it diminishes the rate of catalysis by reducing the proportion of enzyme molecules that have a bound substrate. So the inhibitor constant, Ki, can be defined as the dissociation constant of the inhibitor enzyme complex. That is to say, Ki is equals to product of enzyme concentration and that of inhibitor concentration all over the inhibitor enzyme substrate inhibitor enzyme complex concentration. So the classic example of competitive inhibition is the inhibition of succinate dehydrogenase by malonate and other dicarboxylate anions. So succinate dehydrogenase enzyme is one of the enzymes that is responsible for the reaction of tricarboxylic acid cycle, TCA cycle. You remember Krebs cycle in biology, which is a cycle that's actually completes the production of energy after glycolysis. So the substrates are being furnished to CCA cycle for the completion of the energy production. So it catalyzes the removal of 2 hydrogen atoms from 2 methylene carbon atom of succinate, and the competitive inhibitor malonate resembles, they look like the succinate in having 2 ionized carboxyl groups at pH 7. Beside malonate, other dicarboxylate anide, the oxalate and oxaloacetate may act as inhibitors of succinate dehydrogenase. So if you look at it, the effect of competitive inhibitor can be reduced or totally overcome by increasing. When you increase the substrate concentration, you are reducing or totally overcoming the effect of the competitive inhibitor. So in this type of inhibition, Km usually increase while Vmax it remains the same. Km increases while Vmax remain the same. So this is succinate. Okay, you have succinate dehydrogenase, which is converted to what, which catalyzes the conversion of succinate to what, fumarate, right? So this is the succinate dehydrogenase reaction. So if you look at the structure of succinate, look at the structure of succinate, right? So we have the inhibitors that resemble succinate. We have, look at it, we have malonate, we have oxalate, we have what, oxaloacetate. They are what, other inhibitors of what, succinate dehydrogenase, which are they competitively bind to the active side of succinate dehydrogenase because they look like succinate, right? They look like succinate. So we have uncompetitive inhibition. In uncompetitive inhibition, we said the inhibitor does not combine with the free enzyme or affect its reaction with its normal substrate. So it's uncompetitive. It means the inhibitor does not react with what free enzyme or affect its reaction with the normal substrate. Rather, it combines with the enzyme substrate complex to give us what we call enzyme substrate inhibitor complex, which cannot undergo further reaction to yield the normal product. So what it does, it allows the enzyme to go for its normal substrate to form enzyme substrate complex. Then it will now come and bind the inhibitor, the uncompetitive inhibitor will now come and bind to the enzyme substrate complex, so that it will come now become enzyme substrate inhibitor complex. So the product of the reaction will not be produced. So you see, so it's competitive, it's uncompetitively inhibit the enzyme. So the Ki here is going to be, and its reaction is like this: enzyme substrate complex plus the inhibitor to form the enzyme substrate inhibitor complex, and the Ki, which is given by the product of the enzyme substrate complex concentration multiplied by the concentration of inhibitor all over enzyme inhibitor substrate complex concentration. So we have the Km and the Vmax, they decrease in this type of inhibition. It is common in two substrate reactions. It's of course usually if the reaction is having two substrate. Okay, if it is two substrate reaction, one substrate will bind. Then when the substrate enzyme complex is formed, then before the second substrate bind, then the uncompetitive inhibitor will now bind and form that complex, preventing the production of the product. So we have non-competitive inhibition. So in non-competitive inhibition, the inhibitor can combine with either the free enzyme or the enzyme substrate complex, which interfere with the action of both of them. Okay, so none of the non-competitive inhibitor binds to active site of the enzyme, uh, bind towards side on the enzyme different from the active site. So it doesn't bind to the active site, it's bind to another side that is different other than the active site, often which deform the enzyme. So it does not form the enzyme substrate complex. So when the enzyme is being deformed, and it will not form the enzyme substrate complex, even though it's the enzyme bind to the substrate. Okay, so it does not decompose the normal rate to yield product. So no product reaction did not proceed normally. So these effects are not reversed by increasing the substrate concentration. So in non-competitive inhibition, the reaction with this inhibitor yields two inactive forms: the enzyme inhibitor complex and enzyme substrate inhibitor complex. So this is how it goes. And the Ki is given by KEI, it means K enzyme inhibitor complex is given by the product of the enzyme concentration and that of the inhibitor concentration over the enzyme inhibitor complex. So we have the K enzyme inhibitor substrate complex as the product of the enzyme substrate complex and that of the inhibitor concentration all over enzyme substrate inhibitor concentration, which may be or may not be equal. So here Vmax decrease while Km remains constant. So you should please take note of this little information. Km decreases while Vmax decreases while the Km remain constant. So example of this, we have metal ions, enzymes that require metal ions for their activity by agents capable of binding to the metal ion, e.g. EDTA, ethylene dimethyl triacetic acid. It reversibly binds to magnesium ion and other divalent cations. It's now non-competitively inhibits some of the enzyme requiring such activity. So if we look at the effect of these inhibitors on Lamb-Weber-Buck plot, we say, you know, Lamb-Weber-Buck plot is also called the double reciprocal plot, right? The double reciprocal plot. So this is actually not really clear. Okay, let me show you. Hold on. Let me show you. Okay, let me show you. Excuse me. Let me show you the more clearer one. Okay. So if we look at this, okay, right. All right. Now, if we look at this, these figures, they are more clearer, right? Let me zoom it. Okay. So if you look at these figures, they are more clearer. So this is effect of what? Double reciprocal plots showing the effect of competitive, non-competitive, and uncompetitive inhibition. So if we look at it, this first one is the competitive what inhibition. If you look at this graph, you can see that for the normal reaction, which there is no inhibitor, look at it, no inhibitor. You remember the normal slope is Km over Vmax. You remember the double reciprocal plot, Lamb-Weber plot, and the intercept on the y-axis is 1 over Vmax, right? So this is, you remember, double reciprocal plot, 1 over substrate concentration, 1 over what? Vmax, 1 over what? V. So 1 over V against 1 over substrate concentration. So, but when there is an inhibition, in the presence of inhibitor, you have the slope becoming 1 plus inhibitor concentration over Ki, inhibitor concentration constant, right? All multiplied by Km over Vmax. And the intercept here will become minus 1 all over Km plus 1 Km open bracket, Km will multiply 1 plus inhibitor concentration all over Ki, right? So for that is for competitive. Now for uncompetitive inhibition, you have the without inhibitor, Km over Vmax remain the same, right? So even with the inhibition, because it's uncompetitive, Km over Vmax is still the same. But the intercept, right? The intercept at x-axis is 1 plus inhibitor concentration over Ki all over this negative, right? All negative over Km. That is minus 1 plus is inhibitor concentration over Ki all over Km. And the slope at the, sorry, and the intercept, right, at the x-axis will become 1 over Vmax, open bracket, 1 plus inhibitor concentration all over Ki. Okay, so for the non-competitive inhibition or non-competitive inhibitor, without the inhibitor, normal slope is what? Km over Vmax, right? Right? So what happens if there is an inhibitor? Without inhibitor, this is it. With the slope is what? Km over Vmax. But when the intercept without inhibitor is what? Minus 1 over Km. But if there is an inhibitor, the slope becomes Km over Vmax, open bracket, 1 plus inhibitor concentration over Ki, and the intercept becomes what? 1 over Vmax, open bracket, 1 plus inhibitor concentration over Ki. So you should please take notes of the various differences. Okay, let's go back to our lecture material. Okay, so we can see that it's the same thing, but that's the other one that I just finished showing you is more clear. It's more clear. Okay, so okay, now so we have, this is a summary of the effect of inhibitors on Longuever-Burk plots. You know, we have seen the intercept, the slope, the y-intercept, right? So this is the summary for without inhibitor, with competitive inhibitor, uncompetitive inhibitor, and non-competitive inhibitor. We have already seen them. This is just a table showing this summary. So that is that about reversible inhibition. So now let's look at irreversible inhibition. So some enzymes undergo irreversible inactivation when they are treated with agents capable of covalently and permanently modifying a functional group required for catalysis, making the enzyme molecule inactive. So this type of inhibition cannot be treated by Michaelis-Menten principles. Often the inhibition sets in slowly compared with the normal reaction kinetics of the enzyme, so that the enzyme inhibition is incomplete, so that the inhibition is now incomplete at first, but continuously increases with time because chemical modification of an increasing fraction of the enzyme molecule takes place. For example, a glycating agent such as iodiacetamide irreversibly inhibits the catalytic activity of some enzymes by modifying essential cysteine residues. Heavy metals such as mercury and lead salts also inhibit sulfuryl hydryl enzymes, right? Example, eggs, for example, or egg whites are sometimes administered as an antidote for accidental ingestion of heavy metals. The egg white protein of albumin is rich in sulfuryl hydryl groups, sulfhydryl group rather, sulfhydryl group, which traps the free metal ion and prevent their absorption from the gastrointestinal tract. So in many cases, irreversible inhibitors are used to identify active site residues involved in enzyme catalysis and to gain insight into the mechanism of enzyme action. by sequencing the protein. You remember sequencing that we talk about, knowing the individual amino acid that makes up that protein. So by sequencing the protein, it is possible to identify the specific amino acid residue that are modified by the enzyme inhibitor and which are now involved in the catalysis. So regulation of enzyme activities. How does enzyme activity is controlled? How do we control activities of enzyme? So some enzymes possess special properties which allow them to, which endow them with regulatory roles in metabolism. Such enzymes forms are called regulatory enzymes. There are two types of regulatory enzymes. We have allosteric enzymes and covalently modulated enzymes. So allosteric regulation, we said the catalytic side of an enzyme is modulated through a non-covalent binding of a specific metabolite at the site on the protein other than the catalytic site. So allosteric enzyme, they have another site for binding, for modification, for control, for right, other than the active site. They have a binding site other than the active site. So most enzymes which are subject to such modification are rate-determining enzymes in metabolic pathways. When we say rate-determining enzymes, enzyme that catalyzes a critical step in the reaction pathway. So without that reaction pathway, without that rate-limiting reaction, means the pathway will not proceed. So metabolites that bind to the allosteric side are called allosteric effectors, modifiers, or modulators. These enhance the protein activities. Those that enhance the protein activities are referred to as allosteric activators, where those that decreases the protein activities are called allosteric inhibitors. So the term allosteric comes from Greek word allos and stereos, which means solid subject, in reference to the fact that the regulatory side of an allosteric protein is physically distinct from its active site. We have already explained that. So most allosteric enzymes are oligomeric. That is to say, they consist of several subunits. They consist of two or more polypeptide chain subunits, and usually in an even number. Allosteric enzyme usually exhibit sigmoidal shape, S shape. Their kinetic curve usually is what? Is S shape. So rather than simple hyperbolic curves that we see in representation of what Michaelis-Menten rate equation. So activators of allosteric enzyme shift these hyperbolic and hyperbolic curves, or the S curve, to the left, whereas allosteric inhibitors, they shift the curve to the right. So we have what we call a homotropic allosteric modulator, which is a substrate for its target enzyme as well as regulatory molecule of the enzyme's activity. It is typically an activator of the enzyme. On the other hand, a heterotropic allosteric modulator is a regulatory molecule that is not also the enzyme substrate. It may either be an activator or an inhibitor of the enzyme. So we have end product inhibition. So when we say end product inhibition, in many instances, in which the final end product of multi-enzyme metabolic pathway is an allosteric inhibitor of an enzyme that catalyzes an early and irreversible step of the pathway. So end product inhibition, usually the end product that is being produced by a pathway usually comes and now inhibits a critical step in the pathway. So this form of allosteric regulation prevents accumulation of additional end products and of metabolic intermediate once a cell has a sufficient supply of that metabolic end products. Example, aspartate transcarbamylase, enzyme that catalyzes the first reaction unique to pyrimidine biosynthesis. The enzyme is multi-subunit protein complex which composed of 12 subunits, forming 2 trimers of catalytic subunit and 3 dimers of regulatory subunits. So it is a feedback inhibited by the end product of the pyrimidine pathway, which is CTP, right? So following treatment with mercurials, the enzyme aspartate transcarbamylase loses its activity to the inhibition by CTP, but retains its full activity for the synthesis of carbamyl aspartate. This suggests that CTP is found at a different site other than apart from the different site from the substrate. So this is typical. I don't know if you can see it clearly. So this is in the inhibition of aspartate transcarbamylase. Okay, you have aspartate transcarbamylase reaction. This is aspartate. You have carbamyl phosphate and aspartate. You have what carbamyl aspartate, and you have what we call UMP to UTP now to CTP. So if you look at this, the inhibition here, okay, now the end product CTP now comes, and now if it is now produced an ample supply that the cell does not requires it, it now comes and what inhibit this first step of the reaction. So this is end product inhibition. We have covalently modulated regulatory enzymes. So here the active and inactive forms of the enzyme are interconverted by a covalent modification of their structure, catalyzed by other enzymes. So maybe another enzyme now come and catalyze, maybe remove a substrate or a substituent from the enzyme to make it active, or maybe remove it to make it inactive, or add it to make it active, or add it to make it inactive, as the case may be. So an example is glycogen phosphorylase, which catalyzes the breakdown of glycogen to yield glucose 1-phosphate, right? So the enzyme, this it occurs in two forms: phosphorylase A, which is more active form, and phosphorylase B, which is less active form. So A is converted to B through hydrolytic removal of phosphate groups attached to 4 serine residues contained in each of these subunits, which is brought about by the action of phosphorylase phosphatase. So phosphorylase phosphatase now come and act on the what? By removal of phosphate group that is attached to the 4 serine residue in what? In the subunit. So that is to say, that's simply what happens in covalently modulated regulatory enzymes. So we also have what we call a clinical application. Although there are other ways by which enzyme activity is regulated, and you can say activation of inactive precursors of enzymes, which are zymogens, to yield the catalytic forms, is found in digestive enzyme pepsinogen to pepsinogen, trypsinogen, chymotrypsinogen. These are converted into their active forms by selective hydrolytic cleavage of one or more specific peptide bonds in the zymogen molecule as follows. So there we can have another zymogen, which is an enzyme, but is in its inactive form. So it can be activated to its active form. For instance, pepsinogen can be activated to pepsin, okay? Okay, pepsinogen, which is now, we can have pepsinogen to pepsin and peptides plus peptide, which is now catalyzed by pepsin and so on. You can see we have and trypsinogen also, we have chymotrypsinogen as well. So we have clinical application of enzymes. We can say that many tissues produce enzymes that are relatively cell specific, because these enzymes are released into the circulation as a result of tissue damage. So assays of the level of certain enzymes in blood can provide useful diagnostic information. For example, as ALT, alanine aminotransferase, and AST, aspartate aminotransferases, are present in high concentration in hepatocytes. When these cells are injured, for example, by viral hepatitis or drug overdose, ALT and AST are released into the blood. So several other enzymes serve as tissue-specific marker for cellular injury. So when there is a cell injury, there may be release of some certain enzymes, which when you assay for those enzymes, when you see them in high concentration in the blood, now you can give inference to say that, okay, we are suspecting maybe tissue damage. Okay, we are suspecting so. Now do the diagnosis. So we have what we call also isozymes, which are also very important in diagnosis. These are multiple forms of a given enzyme that occur within a single species of an enzyme, of an organism or a single cell. They differ in amino acid composition and thus in their isoelectric properties. They can be separated from each other by electrophoresis. They also differ in their kinetic parameters and their physical properties. Isozymes with wide clinical application include lactate dehydrogenase, creatine kinase, alkaline phosphatase. Lactate dehydrogenase occurs as 5 different isozymes in other tissues of vertebrates. They all catalyze the reaction lactate plus NAD plus the pyruvate plus NADH plus hydrogen ion. They have the same molecular weight and all contain 4 polypeptide chains. They consist of 5 different combinations of 2 types of polypeptide chains designated M and H as follows here in the composition, the type, the composition, and the reaction where they are found. Lactate dehydrogenase 1 to 5. This is H4, H3M, H2M, HM3, HM4. This is found in myocardium and red blood cells, kidney, brain, liver, and skeletal muscle. Okay, this is an explanation. Okay, so we can say that although they catalyze the same reaction, they differ significantly in their Km and Vmax values for substrate, particularly pyruvate. So they have the same and they catalyze the same reaction, right? But they differ in their substrate affinity. Km is a function of the substrate affinity, right? And Vmax and how fast the reaction take place. So particularly for the pyruvate, which is their substrate. So M4, we can see that it's found in liver and skeletal muscle has a relatively low Km for pyruvate, while H4 that is found myocardium and red blood cells, we can say has highest Km for this substrate. So the other have kinetic properties that are intermediate between those of M4 and H4 isozymes in proportion to their relative content of M and H chains. These characteristics provide insight into the function of the isozymes, and skeletal muscles and embryonic tissues tend to utilize glucose anaerobically and break it down to form lactate. Thus, we can say M4 isozyme is well adapted to convert pyruvate rapidly to lactate. Heart muscle and other, on the other hand, does not normally form lactate from glucose. So lactate dehydrogenase isozymes are important in diagnosis of heart and liver diseases. That is the take-home message. Normally, blood contains little LDH1, but the level increases after myocardial infarction. So this is the diagnosis after it's been noticed. Increase in LDH5 is indicative of secondary liver damage in patient. Thus, measurement of this isozyme allows monitoring of secondary complications of heart failure. And for serum enzyme used in clinical diagnosis, these are the serum enzymes here, and these are the major diagnoses. For AST, myocardial infarction, ALT, viral hepatitis, alpha amylase, acute pancreatitis, ceruloplasmin, hepatosplenicular degeneration or Wilson's disease, and you can see them up to what we call alkaline phosphatase enzymes, which are diagnostic markers for various bone disorders and obstructive liver. So we can say we have come to the end of the lecture. So feel free to read the lecture material, watch the video, and ask questions where you do not understand. Thank you very much. See you next. Bye-bye.